Stagenford, and we are going to be looking at the reason why DNA replicates. So the last couple of days, we've been seeing the complicated process of copying DNA. But a couple of you have actually asked, and with a rightly puzzled expression of why in the world does this even happen? Why would we want to duplicate DNA? And that's a good question to ask, and I'm glad you're thinking that. Um, in fact, if I could scroll down, I actually have a little section here. Why do cells divide? Well, why does DNA replicate? Well, DNA replicates because cells must divide. And then if you follow that line of logic, cells divide because we need to make new cells. And if you continue on, if we need to make new cells, why do we need to do that? Well, we could replace the cells that are gone, like skin cells. They flake off, they die, and then we need to replace them. Or you could skin your knee and then you need a repair. Or just the fact that you're growing. And so when cells divide, they need a copy of DNA as well. So in case this was something that you've not thought of before, if we have our little stick figure person here, um, there's a little rib cage and he's walking around, every single one of those cells has DNA. Well, there are a couple of exceptions. We'll talk about that. So his eye cells, his nerve cells, his skin cells, his bone cells, muscle cells, pancreatic cells, each one of those has an entire set of DNA. Now, there are a couple of exceptions. Red blood cells don't have a nucleus, so they don't have DNA. The muscle cells have more than one nucleus, so they have more than the usual set of DNA. Hair is dead cells, so they've lost their DNA. So there are some kind of quirkiness, but by and large, every cell has its own set of DNA. And that, I hope, makes you think about why that is. Why do cells need DNA? Which is going to be the next thing we look at. So we turn our attention to cell division, and that's called mitosis. Now there have been different ways to try to remember this, but since we talked about growth, you can think about the fact that when you grow, you need to make your body cells, and so over the years we've come up with something like mitosis makes mitose, which is kind of silly, but it works, right? So just the fact that you're growing and dividing and replacing cells. So you want to divide the nuclear DNA. And when you divide that DNA, you're going to separate it, and then you're going to have two equal daughter cells. So you have the same number and the same type of DNA. Now, how is this DNA seen? Well, it's seen as chromosomes. And chromosomes, this is another one of those annoying words where we named it before we really understood what they were. Chromo is going back to the idea of color or dark area, and then som is body. And so what they saw under the microscope were these really dark speckled bodies. So if this is our cell and here's our nucleus, they would see these really dark stained sections inside the nucleus at some point in time, and so they gave them that name. Now we know that chromosomes are primarily made of DNA. Now there are proteins involved, but really that's a packaging idea. So we have chromosomes and they're made of DNA, and the DNA contains genes, and these genes you can kind of think about, they're like a sentence. They mean something. So you have multiple genes in a section of DNA. So you might have gene 1, and then you might have gene 2, and then 3, and all the way maybe up to gene, 100 and, 20, yeah, in a single chromosome. So the gene is a sentence, and then a chromosome would be like the pages in an entire book. And then our entire set of chromosomes would be the book. Now, when you are ready to divide, you need to make an extra copy. So we have our double helix. So see my little red line here, that's representing the other strand, the double helix. If we make a copy, we're going to make another double helix. Now, here would be one, and then they're going to join together in the middle, and the middle is called a centromere, and they have to keep a common convention of how they name these things. So when we have a duplicated chromosome, it's still called a chromosome. So this entire structure of a duplicated chromosome, that's still called a chromosome. But you can think of it as, well, this is a duplicated chromosome. Over here, we are still calling this a chromosome, but this one is unduplicated. Now, because we have two copies, the two copies are called sister chromatids. So the way you would say this, here we'd have one chromatid, here we would have another chromatid. So let me just give you a quick little way to practice. If I look at a group of chromosomes in a nucleus and I'm drawing them out, how many chromosomes do you see there? How many sister chromatids do you see there? Well, there are 6. 1, 2, 3, 4, 5, 6 chromosomes. There are 12 sister chromatids. Now, you'll see for now, just accept the fact that we're going to be working with always the even number of chromosomes, but you'll see why that is in a few weeks. So just to give you an example, humans have 23 pairs of chromosomes. Other animals have a different number of chromosomes. So when we want to get 2 cells, that's where the duplicated DNA comes into play. So again, why are we doing this? Well, we're doing this to replace old cells or cells that have died. We're doing this to repair cells from maybe a cut or a scrape or damage that's been done. And then we're also doing this overall just to grow, for animals to get bigger, for plants to get bigger. Let's not forget the plants. When plants are growing, they're producing new cells. Now, right now, being in the wintertime, it's not, a lot of plants are dormant or at least very slow growing, but they are still going to divide, especially during the springtime and summer. Now, let's wrap things up with a cell cycle. So the cell cycle is how we would view a cell's life. So we're going to start with a freshly divided cell, and a cell that is just newly divided enters a stage called G1. Now, some books like to call this a gap. Some books like to call it growth. And whatever you'd like to think about it, that's fine. Now, this is a time where this is a normal cell function. So if it is a nerve cell, it's busy sending signals. If it is a photosynthetic cell in a plant, it is busy photosynthesizing. If it is an immune cell, it's busy fighting diseases. If it's a skin cell, it's protecting. If it's a flower cell, it's sending off perfume and attracting pollinators. So this is normal cell functioning. Some cells never get out of this, and so we would say that they would enter into a G0 stage. That's a 0, not a go. So basically this means they are stopped. They just stay and do whatever they want to do. A good example of this would be nerve cells. Neurons, they don't divide, and so they are continually doing cell their job as neurons. Now there are a few assistant cells that can divide, and very, very rarely we might see neurons enter mitosis, but by all functional definitions, they don't divide. So that's G1, and a lot of cells will stay in that position. But if they are ready to divide, or if they get signals, and before they go, the signals have to basically say go. So there is a checkpoint here. And the checkpoint, it really is for the cell to basically do an inventory. Are they ready to go? Now, if they're ready to go, they're going to enter a stage called S. And the S stands for synthesis. And you'll see why, and it will make a lot of sense. This is when the cell is going to copy the DNA. So this is DNA replication. So we're going to have our one strand of DNA, and it's going to go into that chromosomal DNA. Now, just remember, this is a double helix here, and I'm just drawing it as a thin line, but so is this. This is a double helix, so is this. So we've copied that double helix. Now that S phase, once you've entered that S phase, the cell is pretty committed, because you don't go forth with the effort of copying your DNA without being committed to dividing. But there are, again, checkpoints where the cell can kind of say, okay, we're ready, we're not ready to go. Then it enters another period of G2. And some students, again, you can think of it as a gap or growth. At this point, we are preparing for cell division. Now you could ask, well, other than copying DNA, what's the big deal? Well, the cell needs to get all of the organelles in place. Do we have enough mitochondria? Do we have enough rough ER? Do we have enough lysosomes? And then we're ready to go at that point. Then there's another checkpoint that allows the cell to do an inventory, because if there are any damage, if there's mutations, then we want to stop and not copy a mutant cell. So there are built-in checkpoints for this. If it's all ready to go, then you enter into mitosis, and that's the cell division, the nuclear division. So we have 4 phases. We have prophase, we have metaphase, we have anaphase, we have telophase, and then cytokinesis, which sometimes is kind of lumped in with telophase. That's when the cell divides, the cell membrane divides. So the next video, we're going to be seeing kind of how this works, what's going on with that. So hope that helps get us started next week, and have a nice weekend. There is a part 2 to this video, so make sure you watch the other one.